my dear students, I welcome you in my channel Eduscope. Today I am going to describe the fourth step of recombinant DNA technology. In my last two videos regarding this RNA DNA technology, I discussed up to the third steps, because you know that there are 10 steps in recombinant DNA technology. In this recombinant DNA technology, first step is selection of the desired character. Second, isolation of the DNA having desired gene. Fragmentation of DNA by enzyme. Isolation of DNA by gel electrophoresis. Amplification of DNA by PCR, means polymerase chain reaction. Ligation of this DNA into a vector from recombinant DNA. Insertion of the recombinant DNA into host. Insertion of recombinant DNA into the host, culture of the host in the suitable medium, extraction of the gene product, and downstream process. These are the 10 main steps for recombinant DNA technology. In these third steps of recombinant DNA technology, with the help of enzyme, that is restriction endonuclease, the DNA is cut into different fragments. And there are fragments of different size. Some are small, some are large. But we want very small DNA fragments having the desired genes. For the separation of the smaller fragments from the larger one, this is the process called gel electrophoresis. Electrophoresis is a technique of separation of charged molecule under the influence of an electric field, so that they migrate in the direction of the electrode bearing the opposite charge through the base material called matrix. Here one base material is required, that is called matrix, and here this matrix is gel. That's why it is called gel electrophoresis. So gel electrophoresis is a process by which electricity is required, some electrodes are required, which attracts the charged particle. As this DNA is having negatively charged, so these are attracted by positively charged anode. That is done in this process, gel electrophoresis. As I told you, as a matrix, gel is used here. There are different types of gels are used in this process, gel electrophoresis. Some are polyacrylamide gel electrophoresis, which is used to separate, that is protein. This is used to separate protein. Next, agarose gel electrophoresis, which is used to separate DNA fragments. So here we are going to describe this agarose gel electrophoresis, because here we are dealing with DNA. And this agarose gel is obtained from agar. This agar is obtained from red algae, and this red algae's example is Gracilaria, or Gelidium. So these are the example of red algae from where agar is obtained. This agar contain heterogeneous acropectin and linear polysaccharide. These are the main component of this agar. This agarose is the purified form of agar. This agarose is the purified form of agar, which contains only this one, only linear polysaccharide. Agarose contain only linear polysaccharide of agar, so it is obtained by the purification of agar. Now I am going to describe this process in detail. This technique of gel electrophoresis was developed by scientist TISELUS in the year 1937. So in the year 1937, TISELUS developed this technique, gel electrophoresis. And here the requirements are C, that is casting tray, or is a chamber, agarose gel, electrode with electricity, ethidium bromide, that is one stain, ultraviolet rays, and DNA fragments. And this is the arrangement. You see, this is a chamber or the casting tray, it is here, and where we have to take this agarose gel. And as I told you, this agarose gel is a chain of polysaccharide, linear polysaccharide chains, and when these chains are present over here, it makes a mess and having small pores over there. Small spores, these are present like this way. These linearly thread particles are present over here, that is agarose gel. And here are some wells through which we can give the DNA fragments. We have to apply DNA fragments through this well. We have to give these DNA fragments. And in this side, one electrode, that is cathode, negatively charged. In another side, there is another electrode, anode, that is positively charged, that is connected with electricity. And here you see that when these fragments are given over here, they will try to move towards this positive. Why? Because you know what DNA particles present over here or given over here, they will try to move towards the anode because this is negatively charged particle. This is negatively charged particle. So DNA is a negatively charged, so they will try to move this towards the anode positively charged. But all fragments cannot move with equal space. All fragments will not move towards the anode with equal pace. Why? Because I told you that this agarose gel makes a mess and makes some pores, and through these pores larger particle cannot move properly, whereas smaller particle can go through the pores which is formed by the agarose gel and it will move towards the anode. And as we cannot see the DNA fragments easily, that's why we have to give one stain, that is ethidium bromide should be given over here, and after that ultraviolet rays should be applied. So see the movement of the DNA particles clearly, we have to apply ethidium bromide and ultraviolet ray, so these particles of DNA will become orange in color. Understood? In the large particles cannot move properly, but smaller particle is moving towards the anode, so it is orange in color. This will change into orange. So it will be easier for the scientist to pick it up, to see the location of these smaller fragments. After a stipulated time, when scientists come to know it is separated, then this type of banding of the DNA is compared with a standard chart, that is called DNA ladder. What is that? DNA ladder is the standard chart, and this type of banding or separated DNA fragments is compared with the DNA ladder, and they come to know which fragments is ideal. When scientists come to know the ideal length of the DNA, these are ready to be taken out. So agarose gel is cut and these small fragments is taken out from this gel electrophoresis setup, and this process is called elution. Cutting of agarose and extraction of DNA from this setup. Gel electrophoresis setup is called elution. So elution is a very important process. You see, elution is an important process by which this agarose gel is cut and the smaller fragments of the DNA or carrying the desired gene is to be taken out. That is called elution. After this process of elution, when DNA fragments is taken out, that is to be amplified by the process of PCR, that is polymerase chain reaction. And the process of polymerase chain reaction will be discussed in my next video. So if you want to see that video, subscribe my channel Eduscope, and thank you very much for being with us for a longer time.